Okay, so this topic is all about the red blood cell anomalies or abnormalities. Okay, for the classification here of the abnormalities related to our red blood cells. So number one, we have here abnormality in terms of the size. You call the transfer anisocytosis. The second one, we have here abnormality with the hemoglobin content. You call this one your anisochromia. Abnormality in the shape. So we have here the poikilocytes. And we have also here the presence of the different inclusion bodies. So this topic we're going to discuss here, the morphology, abnormalities, and the related medical condition to each of the different abnormalities. Okay, first we have here the abnormality in the size. We call this one anisocytosis. Okay, for the basis here to classify our red blood cell size in terms of the anisocytosis. So we have here the blood cell index in the form of the MCV or the mean corpuscular volume. Okay, the normal reference range for the MCV is 80 to 100 FL. So based on that reference, so if the MCV result of the patient's sample here is between 80 to 100 FL, so we consider here the red blood cell of the patient to be normocytic. So normocytic could be found here in the normal red blood cells. Could also be found here in our normocytic anemia that includes our hemoglobinopathies. And we have also here our aplastic anemia. On the other hand, if the MCV value here is less than 80 FL, then we consider that was our microcytic or microcyte. So medical condition associated here with the decreased MCV values. So we have here the chronic post-hemorrhagic anemia. Then we have also here the iron deficiency anemia and thalassemia. Okay, so since in the case of the iron deficiency anemia and thalassemia, they have the same classification as to their MCV value. So we can use the RDW or the red cell distribution with as a parameter to differentiate the two conditions. So if we are trying to take here the normal reference range for the RDW as 11.5 to 14.5%, so patient with the IDA would have an increased red cell distribution width. Whereas in the case of the thalassemia, it would have here the normal RDW result. On the other hand, if the MCV value of the patient here is more than 100 FL, then we consider that one as macrocytic. Okay, macrocyte is seen here in the following medical conditions. So number one, we have here the megaloblastic anemia associated with vitamin B12 deficiency anemia that may be caused here by pernicious anemia or PA. There could also be a sprue, gastrectomy, or even the dilatum infection can induce or cause here our vitamin B12 deficiency anemia. Another one, megaloblastic anemia here would be our folic acid deficiency. To correlate here in the case of your megaloblastic anemia, so it also should be accompanied by hypersegmented neutrophil. So it would have here the presence of the neutrophil having more than 5 lobes. And at the same time, the presence of the inclusion body in the form the Howell-Jolly bodies could also associate with our megaloblastic anemia. Of course, it should be accompanied with MCV value of more than 100 FL. On the other hand, macrocytic could also be found here in the non-megaloblastic condition that includes here the liver disease, hypothyroidism, chemotherapy, and we have here the alcoholism. Okay, so these are the normal red blood cells in terms of the size. Then if we compare that one with this one, so this is macro size, sobrang laki nito. Okay, that's where we have here the macrocytes. And we have here the hypersegmented neutrophil. So this one, a neutrophil more than 5 lobes. That's associated with the megaloblastic anemia. Of course, it's also accompanied with Howell-Jolly bodies. Okay, the next abnormality here, we have the abnormality in our hemoglobin content. You call this one as anisochromia. Okay, to evaluate here, to classify the cells as to hemoglobin content or the pallor area. So we have here another blood index in the form the mean corpuscular hemoglobin concentration. Normally, you have that 31 to 36 grams per DL in terms of the MCHC result. And the hemoglobin should also occupy here 1/3 of the pallor area. Okay, so if the result here less than 36 grams per DL, then we consider that one as hypochromic. So medical condition associated here with the hypochromic red blood cells, same also with the condition related to your microcytic. That's why we call this one your hypochromic microcytic. So that includes, again, our iron deficiency anemia, and we have also your thalassemia. So if there's a here, however, it's more than 36 grams per DL, then we consider that was to be hyperchromic. So most likely hyperchromic may be seen here in our spherocyte. The spherocyte, what happened to that? The normal or the volume of the cell here still remain to be normal. However, the surface volume here or the surface area decrease. That's why we consider the spherocyte as decreased surface area volume ratio. Okay, because of that, it appeared that your red blood cell or the spherocyte appear to be having dark colored throughout the entire cell and would have here the absence of the pallor area. And therefore, it appears to be hyperchromic. So spherocyte can be seen here in our hereditary spherocytosis. It could also be seen here in our autoimmune hemolytic anemia. So differentiate the two. So both of them, that's the cost to try to consider our spherocyte. In the case of the hereditary spherocytosis and autoimmune hemolytic anemia, both of that will have an increased osmotic fragility test result. To differentiate the two here, so we have your antiglobulin test. First, antiglobulin test will detect here our sensitization related here to our autoimmune hemolytic anemia, but not with the hereditary spherocytosis. Okay, for the result here of the MCHC between 31 to 36 grams per DL, so we consider that one to be normal. So it's also seen in our normocytic anemia, so it becomes normocytic, normochromic. So example for that, we have your hemoglobinopathies, and another one is aplastic anemia. Okay, so we have here the hypochromic cells, so it appears to be pale staining. And this is our spherocyte, so it appears to be a full red cell. So the entire cell here is being colored, and there's absence also of the pallor area. Okay, the next abnormality, we have here abnormality in the shape. You call it as your poikilocytes. So if the red blood cell would have here abnormality in the shape, so it would have here that poikilocytes or poikilocytosis, it would have here decreased result with the ESR. So bakit ma-decrease ang ating poikilocyte in terms of the ESR? Because pag poikilocyte ang red blood cells natin, it would have here the inability to undergo rouleau formation. So we need to remember here that the first step in the ESR is rouleau formation. So kung ang red cells natin hindi siya mag-undergo ng rouleau formation, so therefore because meron siyang abnormality in the shape, so nahihirapan siya mag-undergo ng setting or sedimentation with our ESR. So with that, we are expecting that the result of the poikilocytes would have here a decreased result or mabagal na sedimentation. Okay, the next one, we have here the abnormality in the development. So we have here your megaloblastic anemia, and again, that includes your vitamin B12 and the folic acid deficiency anemia. But it was being called here asynchronous development because you have the problem or defects in the DNA if you have your megaloblastic anemia. So there's a problem with maturation of the nucleus. So you have here a normal maturation of the cytoplasm of the cell, but not with the nucleus. That's why we call this one as our asynchronous development. Okay, the next one, abnormality in the RBC membrane. So dito nagkakaroon talaga ng kakaibang shape sa membrane. Okay, the first one, acanthocytes. So other name for that one is your thorn cells per cell. Okay, acanthocyte is our red blood cells wherein the projections are pointed, and at the same time that one would have an irregular projection. Okay, acanthocyte usually seen here if you have the problem with the LS, lecithin sphingomyelin ratio. Of here the case of the beta proteinemia, and even associated with our pyruvate kinase deficiency. Echinocyte, or other name for that was Burr cell, sea urchin, or kinated cell, it will resemble also our acanthocyte. However, the projection here is evenly distributed. Unlike with acanthocytes, so the projection are not even. But in the case of the echinocytes, this one would have an even distribution of the projection. Okay, kenoside here associated with patient with renal problem. And also, that can also induce the red blood cell to become cleanated as well. If you're going to collect the blood and the ETA and then store that one for a longer period of time. Because the storage, it lost its ADP, and that can result here to the formation of cleanated cell or echinocyte. Okay, then we have also here the target cell. So other name for that one is Mexican heart, codocyte, leptocyte. Okay, so target cell or Mexican heart here characterized by the red cell with a central pallor area surrounded by a clear staining area. That's why, because of that, it appears to be a target. It's a target cell because of that. Okay, the target cell or the Mexican heart here would have an inverse or opposite description with our spherocyte. So in the case of the target cell, what you have for that is increased surface area compared to the volume ratio. Okay, the target cell always associated with the thalassemia. So pag nakakita tayo ng target cell, so we're suspecting here for the thalassemia as our medical condition. Target cell can also be associated here with the hemoglobinopathies natin. Okay, then we have here the spherocyte. So spherocyte characterized here by decrease or deficiency in the protein spectrin. So this one is characterized here by the normal volume but decreased surface area. Okay, so spherocyte can be seen here in our hereditary spherocytosis. Okay, leptocytes, on the other hand, here is our bipolar hyperaggregation of the hemoglobin. Okay, so that's primarily related to the defects in the cytoskeleton. Okay, then we have here the stomatocyte or mouth cells. So this is our slit-like, mouth-like pallor area. So para siyang mouth-like at the central portion of the red blood cell natin. Okay, stomatocyte or mouth cell associated here with the RH null. So pag sinabi natin RH null, so you don't have here the antigens related to RH. You don't have the antigen for the C, the E, and the D antigen, which are all related to our RH. Stomatocytes also shaded here with abnormality with our electrolytes. So electrolyte abnormality here that lead to stomatocyte or mouse cell is characterized by an increase in the sodium tapos bumababa si potassium natin. Because normally dapat mas mataas si potassium inside the red blood cells natin kaysa sa potassium sa sodium natin. Because we consider the potassium as our major intracellular cation while our sodium is our major extracellular cation. However, here in the case of the stomatocyte, so mas dumadami ang concentration ni sodium sa loob ng red cell natin compared to the potassium. And that lead here to our stomatocyte or mouth cell. Okay, the next one, we have here the presence of the abnormal hemoglobin. So, number one, meniscocyte. Other name for that one is our drepanocyte. Meniscocyte, drepanocyte here is our sickle cell. This one characterized here by the red blood cells which is transformed or convert to a holly leaf or letter S or letter C na shape ng red cells natin. And we consider that one as a reversible transformation. So once it try to transform to become sickle cell, it can no longer already return back to the normal biconcave disc. So with that, it would increase the sequestration of spleen, leading to the hemolytic reaction. Another one, we have here the presence of your trauma that may be related to physical, chemical, or mechanical trauma. So pag traumatic injury, so we are suspecting here for the presence of the keratocytes or the horn-like. We have also here the mesocytes, or we have also here the schistocytes. Okay, usually this one, nagkakaroon sila ng physical trauma because of the red blood cells has been caught in the fibrin strand or altered vessel, like in the case, for example, the microangiopathic hemolytic anemia. So pag may situation or question related to what's the morphology of the red blood cells which has been subjected to trauma, so ang sagot natin ay schistocytes. On the next one, we have here the teardrop or the cryocyte. So ito ay related sa ating MMM or myelofibrosis with myeloid metaplasia. Okay, then we have here the pyropoikilocytes. So this is characterized here by the extreme sensitivity of the red blood cells in the temperature. Usually, the red blood cell try to fragments and tries to subject it want to hit at 49 degrees Celsius. Pero ang pyropoikilocytes, so nagpa-fragment na ito kahit ang temperature natin ay 45 degrees Celsius pa lang. So kahit hindi pa siya umabot ng 49, so being extra sensitive, so kahit lower pa ang temperature natin, nagpa-fragments na siya kaagad. Okay, the next one, we have here the presence of the inclusion bodies. Okay, so number one, under that, we have here the Howell jelly body. Okay, the Howell jelly bodies is considered to be our fragment of the DNA. So this is the defects related here to the nuclear maturation. Howell jelly body is associated with our megaloblastic anemia. So that includes your folic acid deficiency and your vitamin B12 deficiency anemia. So alongside with hypersegmented neutrophil, the presence of the Howell jelly body signifies here our megaloblastic anemia. Okay, so in the picture here, this is our spherocyte. So kasi sobrang colored or full ang red blood cells natin, wala siyang pallor area. So that's our spherocyte. Okay, this is our mouth cell or the somatocyte. Again, this one associated here with the RH null or the electrolyte abnormalities. Okay, this is our mouth cell pa din. Okay, then we have here the eleptocyte. So this is our elongated. Again, the eleptocyte here associated with hereditary eleptocytosis. That one associated here with abnormality in the cytoskeleton. Okay, we have here the dacryocyte or the teardrops. So that's one associated with our MMM or the myelofibrosis with the myeloid metaplasia. Okay, we have your schistocyte and schistocyte. Again, this one is related here to the physical trauma. Okay, pag traumatic injury, so that's result here the presence of our schistocytes. Okay, then we have also here our sickle cell. So this is just your other name for that one is your meniscocyte and drepanocyte. Okay, so again, this is an irreversible conversion of the red blood cell here to the letter S or the letter C and or the sickle cell. And once the red blood cell here is already converted to our sickle cell, it can no longer be able to return back to the normal biconcave disc shape. What trigger here the sickling process is the decrease in the oxygen supply in the body of the patient. So like if the patient suffering with the dehydration, so mababa ang oxygen, so therefore it try to allow the sickling process. Okay, we have here the target cells. So this is your Mexican hat or podocyte, leptocyte. So this one is associated with our thalassemia. Okay, so this one is characterized here by increased surface area volume ratio. And also, target cells also associated here with hemoglobinopathies. Okay, now we have here other inclusion bodies. So we have here the Cabot ring. The Cabot ring is just a ring or a loop. And this one is the fragment or the remnant of the mitotic spindle. Okay, the Cabot ring is associated here with our severe anemia. Okay, the next one, basophilic stippling or the punctuate basophilia. So this one associated here with our lead poisoning. So usually the patient here would have this basophilic stippling because of the absence of the enzyme pyrimidine 5' nucleotidase. So that's the enzyme that's involved in the catabolism of the RNA. So since the patient don't have that enzyme, so the RNA remains to be uncatabolized, and that become here the fragment of the RNA, and that become here your basophilic stippling or the punctuate basophilia. Okay, the next one, Heinz bodies. Okay, the Heinz bodies here signifies our precipitated globin, and that's associated with the defects or the problem here in our hexose monophosphate SHAN pathway. In the hexose monophosphate sham pathway, we are needing the enzyme glucose-6-phosphate dehydrogenase. So, pag wala kang enzyme na glucose-6-phosphate dehydrogenase, so eventually you will not able to produce here the most important product of this pathway, which is your NADPH. Okay, NADPH is needed in our reductive biosynthesis that provides the energy for our oxidized glutathione to become reduced glutathione. So it's because our reduced glutathione is our functional state ng glutathione that provides us with antioxidant property. So pag wala kang glucose-6-phosphate dehydrogenase, so you do not produce the NADPH. Pag wala kang NADPH, so eventually our glutathione has been already utilized because it tried to exert its already its antioxidant property, you could no longer able to recycle. And because of that, pag wala tayong glutathione, once we're exposed to the oxidizing agent, it results here to the oxidation or precipitation of the globin. So that become our Heinz bodies. Okay, Heinz bodies can be identified by the acetyl phenylhydrazine plus our crystal violet. Okay, then we have here our iron granules. So we have the two siderocyte and we have here the Pappenheimer bodies. Siderocyte or sideroblasts characterized here by the iron granules primarily made up of the hemosiderin. That's why it's being stained here by the Prussian blue stain. Pappenheimer bodies, on the other hand, is another iron granules. That one, however, is made up of the ferritin. Okay, this one can be visualized by the right stain, but not with your siderocyte or sideroblasts. Another one, hemoglobin H. This one is our precipitated hemoglobin H associated with the patient with the hemoglobinopathy na hemoglobin H, or sorry, na thalassemia, which is the form of your hemoglobin H. Okay, hemoglobin H characterized by the presence of your 4-beta globin structure. Okay, then we have here associated with our hemoglobin C. So we have here the CC homozygous. So we have the crystal associated with that. We have your hemoglobin CC crystal. This one is characterized here, being described as the rod-shaped hexagonal crystal with a blunt end, but do not go beyond the red blood cell membrane. Unlike our hemoglobin SC crystal, so this one's your Washington Monument. This one, however, here try to go or try to protrude beyond the red blood cell membrane. Okay, the next one, we have here the abnormality in the distribution. So we have the rouleau formation. Okay, rouleau formation here is being triggered by an increase in the plasma proteins. That's in the case, for example, in the multiple myeloma, Waldenstrom macroglobulinemia. So this just considered to be our false or pseudo-agglutination reaction. So how that one is being manifested as a stack of coin arrangement for the red blood cell. But we need to consider that one is not a true agglutination reaction. So you can disperse the red blood cell here by saline washing. Okay, the next one, red cell agglutination. So this is considered to be a true agglutination reaction. And it's being caused here by our antibody, autoantibodies. Associated with the red cell agglutination, we have the condition called agglutinin disease, called agglutinin syndrome, and we have here the primary atypical pneumonia associated with our cold agglutinin disease. So that one is characterized here by the presence of the cold autoantibody. Pag ganyan, pag red cell agglutination is considered to be a true agglutination reaction, so hindi na natin siya ma-wash off even you perform here the saline washing. Okay, miscellaneous here, we have your polychromasia. So, polychromatophilia, that's polychromatophilia, so mean to say, iba-ibang color ng ating cells. So, that one actually represents our reticulocyte. So, if we're going to stain this one with our new methylene blue, so it will identify it as a reticulocyte. Pero pag right stain lang, so it would appear to be having a variation in color. That's why you call it your polychromasia or polychromatophilia. Of course, sa yung pag-increase sa reticulocyte natin, it will signify our increased production of the red blood cells in response. of our bone marrow to the hemolytic reaction in our body, like your hemorrhage or any other bleeding manifestation. Okay, so here the picture, we have here the Cabot ring. Okay, this is just your ring or a loop. Okay, the Cabot ring is the remnant of your mitotic spindle and is associated here with your severe anemia. Okay, these are your malarial pigments. These are also our steeplings associated with the different malarial infection. Okay, we have here the presence of the nucleated red blood cells. So we need to correct here for the WBC count natin. So when are you going to correct here for the presence of nucleated red blood cells? So pag ang patient natin ay young population or children, so you need to correct here for the WBC count in the presence of your nucleated red blood cells because it's falsely increased the WBC count. So for that, if the patient here is children, so you need to correct that one if you happen to encounter more than 10 nucleated red blood cells. But in the case of the adult population, so you need to correct here for the WBC count only when you're able to encounter here more than 5 non-nucleated red blood cells. So for our formula for that, so we have here your uncorrected WBC count multiplied by 100 over the number of the nucleated red blood cell plus 100. Okay, lastly, we have here the composition of the red blood cell membrane. So the red blood cell membrane, 50% of that's made up of the protein. So there are two types of the protein here. We have your integral and peripheral. Integral protein that's includes here our glycophorin A, glycophorin B, that's responsible here for the negatively charged particles in the red blood cell membrane that will enable that to repel one another, or the salicylic acid. Okay, peripheral protein, on the other hand, example for that, we have the spectrin. So if you have deficiency spectrin, then it can lead to our spherocyte. Then we have also here hereditary leptocytosis characterized here by the abnormality in our cytoskeleton. On the other hand, lipids here accounts for 40% of the red blood cell membrane composition. Okay, for the lipids naman, so abnormality associated with that, so we have your abnormality in your LS, lecithin sphingomyelin result here to our acanthocyte. And the remaining 10% here of the red blood cell membrane is made up of our carbohydrates. Okay, so that's the end for this discussion for this topic. Okay, thank you.